Good morning everyone. So today let's start the next topic, the chromosome. Now, as we know, chromosomes are considered as the hereditary vehicles. Okay, these are the DNA protein hereditary structures which are formed by condensation of chromatin fibers. That means already we have studied about the structure of nucleus. We have found that within the nucleoplasm the chromatin fibers are present, and then also I have told you that they condenses to form the chromosomes. Now these are actually rod shaped or thread like structures which are deeply stainable. Okay, now it was first discovered by Welder. Okay, during his studies he observed the chromosomes. Now actually, the chromosomes normally in somatic cells they occur as diploid cells. That means two pairs are there. But in gametes, that is the reproductive cell, it always occur in the singular form. Okay, it is always occur in haploid condition. Now, the chromosome it consists of two structures, two fiber or thread-like structures which are known as the chromatids. Okay, these chromatids are held together at a point which is known as centromere. Now, within these chromatids, actually numerous fibers are also present which are known as chromonemata, and those chromonemata are held with the help of chromomeres. Okay, now sometimes what happens, now this centromere we can also call it as the primary constriction. Now besides this primary constriction, the chromosome consists of a secondary constriction which we call it as satellite, and the chromosome which bears the satellite is known as the set chromosome or marker chromosome. That means if this particular structure is present, then it becomes easier during karyotype formation or ideogram studies or chromosome studies to identify the particular chromosome. That's why this is used as a marker chromosome. Now, based on the position of centromere, the chromosomes can be divided into four types. Now, if the centromere is present at the center, then we call it as metacentric. That means on both the sides of the centromere, the arms are equal. Now, these we call them as arms, so these arms are equal. In submetacentric, the centromere is submedian. That means in 1/4 position it will be present. Okay. In acrocentric, the centromere is towards the end, and in telocentric, the centromere is at the tip portion. Okay. So based on that, we find four types of chromosomes: metacentric, submetacentric, acrocentric, and telocentric. Now, as these are hereditary vehicles, that means they will help in carrying of genetic information, isn't it? DNA. Whatever is being transmitted from one generation to next, that particular informations are carried in these vehicles. Now, chromosomes are of two types. Okay, these we call them as the giant chromosomes because they are much larger than the normal size of chromosome. And there are two giant chromosomes. One is the polytene chromosome and other is the Lamb-Rush chromosome. Now, the polytene chromosome, it was first reported by E.G. Balbiani in the year 1881 in the salivary gland of insect, and for which this is also known as the salivary gland chromosome. Okay, now this reaches a length of about 2000 micrometer, and this is actually formed by the somatic pairing between the homologous chromosomes. Okay, that means during the pairing state they do not separate, they pair together and then replicate, and for which they attain a large size. Now these particular chromosomes, they bear some swellings which we call them as the Balbiani rings or chromosomal puff. Then they possess some bands, dark bands, and in between the dark bands interband regions are present. Now the puff which are present or the Balbiani rings, here the DNA strands uncoil and produce a large number of mRNA. That means in this position large number of mRNA are being produced, which helps in formation of the further processes, that is in transcription, translation processes also they will now help. And this mRNA, it remains temporarily stored in the puff, and once the processes are completed, this puff again it goes to the normal size. Okay, whenever it is necessary, it bulges out and forms the bulbine rings, and whenever not needed, it then goes to its normal size. Now the second type of giant chromosome is the lamb brush chromosome. This chromosome was first observed by Fleming, okay, but it was described by actually Rucker. Now these are highly elongated special kind of synapsed mid-prophase or diplotin chromosome which have already undergone crossing over. That means they occur in the diplotin stage of most of the animal oocytes during in the actually embryonic stage, okay, egg formation part. They are much larger than the polytin chromosome. So if question comes the which is the largest chromosome, then definitely we can mention that the lamb brush chromosome is the largest chromosome. Okay. Now here what happens actually, the chromatids, they bear a large number of chromomeres which gives out some loop-like structures, and due to this the structure of the chromosome appears like that of the lamb brush. Okay, the brush with the help of which we clean the lamb, isn't it, or the test tube. So it appears like the brush, and that's why the name was given lamb brush chromosome. Now the the loops which here, it consists of several transcriptional units for which it helps in rapid transcription of mRNA. The RNA that is synthesized it starts from the thinner position to the thicker position, and here what happens, a large number of biochemicals are produced which are very much essential during the early development of the embryo. Now after the processes are completed, similar to that of the polytin chromosome, these loops are withdrawn. Okay, when the work is done, transcription is completed, that means the loops will be withdrawn and it will take the normal shape. So this is all about the chromosome part. Now let's just once recall what we have studied. First of all, we have studied the structure of chromosome, which consists of two chromatids attached with the help of a centromere. It consists of the secondary constriction, the satellite, which we call it as the set chromosome or a marker chromosome. Then based on the position of centromere, we have found 4 different types of chromosome: metacentric, submetacentric, acrocentric and telocentric. Then we have got 2 types of giant chromosome. One is the polytin chromosome, which is also known as the salivary gland chromosome, and other is the lamb brush chromosome. Okay. Now remember one thing, in polytin chromosome the puff consists of RNAs. Similarly, the loops also, it is rich in RNAs. Okay. So this is all about the chromosome part. Hope the video is clear to you. Go through it. If you find any problem, then please let me know.